Hi, everyone, and welcome to today's episode of Seed Speaks. This is a video series brought to you by Seed World Group. I'm Mark Zinkowitz, and I'm an editor with Seed World Group. And this month, we are talking with experts about the future of plant breeding and related research. I'm joined today by three young scientists who are doing some groundbreaking research in some very specific areas. Fusarium damage detection in seed, pod shatter in canola, and sequencing the genome of sclerotinia. These three scientists are the winners of the 2023 Canadian Plant Breeding Innovation Scholarships. These scholarships go to deserving young plant scientists who represent the future of our discipline. And this year's winners are Laura Garcia of McGill University, Rajbir Kaur at the University of Manitoba, and Sheila Andrade of the University of Saskatchewan. Welcome, everybody. How are you today?
Thanks, Mark. We are good.
Thank,
What about you?
thank you. We are good.
Good.
We're
Glad.
just excited.
Yes, well, and congratulations to all three of you on your scholarships. You are very deserving. And I want to take a moment to as well thank our sponsors this year. These scholarships wouldn't be possible without the contributions of our sponsors. And this year we have Alberta Grains, Canadian Seed Growers Association, FP Genetics, High Tech Production, Richardson, Sasquatch, Seacan, Seeds Canada, Seed World Canada, Warburton's, and the Western Grains Research Foundation. So thank you very much to all of them for their sponsorship this year. Now, like I say, you are all 2020, I think I said 2023. I should have said 2024 Canadian Plant Breeding Innovation Scholars. And you truly represent the future of plant breeding. You're all working on either a master's or a PhD, and you each study some really unique areas that impact specific crops. And I want to tell our audience why your research is so important. But before we discuss the intricacies of your research, I want to give people an idea of why young people today, like yourselves, are getting into plant science in the first place. I'm going to start with you, Sheila. In a nutshell, can you just tell us a little bit about what inspired you to be a plant scientist, Sheila?
Yes, so thank you for the opportunity to be here and to talk about my project. First, I would like to talk a little bit about myself. My name is Sheila. I am from Brazil, and I was born and I grew up like in a small city where the agriculture was like the main economic activity. So since I was a child, I have been in contact with plants, and this contact triggered me to wanted to understand more about plants. So I wanted to understand their life cycles, how they respond to the environment. I want to like help the farmers to produce more in efficient a way. And in addition to that, I always like the plant pathology. Like inside plant science, I always like plant pathology. I always like to associate a symptoms with a pathogen and how I could like manage a certain disease. In addition to that, like the possibility to help farmers to produce more and in the food safety inspired me to be like a plant scientist.
Laura, I'm curious, what made you want to be a plant scientist?
Thank you. Thank you so much for this opportunity as well. And I guess just similarly to Sheila, I think my interest for agriculture stems from growing up in a small town in my home country in Mexico, where I got to see closely since I was a child how people within the community would work the land, they would grow their own food to support their families. So that was a lifestyle that my own family was following as well. So I really just got passionate about it, and I really wanted to learn more about the resources that we had within the community, but we didn't really know how to exploit them better. So that's when I decided to enroll myself into a plant science program. which then got me acquainted to a plant breeding program through a professor who was doing a research project involving the breeding of sorghum varieties to make them resistant against frost. So that's pretty much when I got interested by all this plant breeding world. And this time I'm still involved in the plant breeding aspect. Just this time I'm studying plant pathogens, which is also an exciting field for me.
And last but not least, Rajbir, what made you want to do plant science?
Yeah, so firstly, thanks for having me. I have similar background as Sheila and Laura. I was born in Punjab, India, which is a major agriculture producing state in India. Like farmers there mainly grow staple food crops like wheat and rice. So like they spend their money, time, and hard work in the fields. But I saw in return they were only able to they were facing a lot of economic losses, and they have had the impact of climate change, diseases, and pests. So I felt it was very harsh on them. On the same way, when I was young, I was very interested in science and the principles behind it. So also I was motivated by reading the Green Revolution, how it happened, and how the scientists in India helped to take it onto another level. So combining all these aspects, I think anyone would land up into plant science.
Now, I want to talk about the research that you all are doing. I'll go in reverse order this time. So, Rajbir, your research focuses on enhancing pod shatter resistance in canola using CRISPR-Cas9 technology. I'm sure most of our audience knows, but in case they don't, pod shatter is a natural process in canola where the seed pods split open during the growth phase, which can pose challenges during harvest due to uneven maturity. So, the goal of your master's research is to develop shatter resistant canola varieties through gene editing, which would allow for more uniform crop yield and quality. Why is this so important, and what progress have you made so far in your work, Rajbir?
So, as you said, Marks, shutter resistance canola is important to maintain uniformity and to reduce the losses from the crops. So, the main reason is we develop hybrids which are shatter resistant and which the breeders in the public universities can use. Although there are many hybrids available from companies which are shatter resistant, but they impose royalties and they have licensing restrictions. So the public breeding programs in the public universities cannot use it. So developing a shatter resistance canola will help a public breeding program like hyaluronic acid rapeseed program here in the University of Manitoba to use this and further dwell into plant breeding. Uh, then I would like to tell about the progress of my project. So initially we transformed uh, two genotypes of canola, which is Brassica napus, with our CRISPR vector, and then we got T0 seeds, T0 generation seeds. We tested hundreds of T0 generation seeds, genotyping them with PCR, and luckily we got positive mutants, positive for the Cas9 insertion. So now we are on the stage where we are waiting for plants to get mature so that we can characterize them, like phenotypic characterization, to know how they have incorporated the gene and are how much shatter tolerant are they as compared to the wild types or as compared to the other Brassica napus plants.
Hmm. Now, Laura, you're pursuing a master's degree on a project. You're working on a project within the Canadian Sclerotinia Initiative, and you're studying a pathogen that affects multiple crops. Now, this project involves genetic diversity studies with a focus on sequencing the pathogen's genome. Now, your goal is to gather valuable genomic information that you can incorporate into breeding efforts. How does your research, Laura, stand to help our industry, and what can you report so far?
Yeah, thanks, Mark. I think the work we're doing in our lab is quite important for the industry because now we managed to get a data set of some of the samples of isolates that are currently affected in commercial fields. And that was thanks to our collaborators and the farmers as well, who helped to collect the samples. So now we have an idea of what's the current population in 3 Canadian provinces in Ontario, Quebec, and Alberta. So that's quite important for us to have knowledge of the population that we're working with. So not only that, we have the data set, but also we managed to gather some information that is really important, such as we were able to phenotype the pathogen into different aspects. First of all, was to characterize them according to the Mycelia compatibility group that they belong to. And also, we now know what is the level of pathogenicity that all these samples that we collected have. So that's pretty important because sometimes we use this information, or plant breeders use this information to choose between the most aggressive isolates when they want to do the screening for the plant breeding programs. So in this case, we're working with common bean. So as of to date, there's only moderate resistance against this pathogen. So by incorporating this valuable information, which is the phenotypic aspects of white mold, we're helping to give a step forward to that. Not only that, we also in our lab managed to sequence the whole genome of all the samples. So we're now trying to explore, we already know some phenotypic aspects, so now we're trying to delve into the genomics aspect of it. So now that we have separation of groups and we know kind of the mode of reproductions of these samples that we collected, we're now looking into some genomic areas to see what are the differences into, more into the genome aspects of the pathogen. So that's going to be pretty important for incorporating this information into the breeding programs. And that's pretty much how we are in with Canadian Sclerotinia Initiative contributing to plant breeding.
Now, Sheila, your PhD research is meant to assist breeders and enhance their understanding of the genetic foundations of Fusarium head blight resistance. Now, your goal is to contribute to the improvement of food safety, reduce agricultural losses, and ultimately enhance the quality of food products. How can all that be done by unraveling the genome of Fusarium head blight, and what can you report so far? Oh, you might be on mute still.
Sorry.
There we go.
So Fusarium head blight, it is a major disease in Western Canada. It reduces like the yield and the quality of the grain, which leads to the development of Fusarium damaged kernels. These kernels, they are like a grading factor. And besides that, we have the production of mycotoxins. These mycotoxins, they are dangerous for human and animal consumption. So unraveling the genome of Fusarium or of wheat, like mainly wheat, we can understand like which genes are related to the resistance. We can understand like the virulence of the pathogen of the Fusarium species, and also we can understand the interaction, like the host pathogen interactions. So far, like some genetic studies, they reported some, from the wheat side, they report like some quantitative trait loci, which are related to the resistant. However, we don't have like a wheat cultivar that is totally resistant to Fusarium head blight. This is why this is because the resistance is related to too many genes, and also like the phenotyping, which is the detection of these Fusarium damaged kernels. It's a long process, and sometimes it's not accurate. And besides that, like the disease, the infection, it's very influenced by the environment. So what I'm trying to do with my project, we are trying to improve improving the phenotype of the Fusarium damaged kernels. In this way, if we get like more accurate phenotype, and we can get like more genetic studies, and also we can improve like food safety.
Now, I'm curious to know some of the challenges that you face in your work. Now, your research can create new opportunities for the seed industry and the people who grow these crops. And you've all been really successful in what you do, but every scientist, of course, faces hurdles that they need to get over. Starting with Laura. Laura, what's the biggest challenge that you face as a plant scientist? And how do you suggest that we as an industry can help to change that?
I would say most of the plant scientists I know, including myself, at some point of their research project, they had faced the lack of adequate facilities to perform their project. So I would say a combination of financial support and maybe fostering more collaboration between the industry, the private sector, and the academia would be a great start to tackle these kind of problems.
Rajbir, how about you? What's the biggest challenge you face, and how can our industry, the seed industry, how can we help you with that?
I think I agree with Laura. I have seen myself and many people land in the space where we, face a problem with lack of facilities, resources, and mainly funds. So I guess industries can really help in providing cost-effective and new technologies like high throughput phenotyping and genotypic techniques to the students. So I think in this way they can really support them. Also, this I get that many students who suffer after completing their graduate programs, they face a lot of problems in finding jobs, like who really want to indulge in industries. So I guess industries can collaborate with academic institutions, and like they can give lectures on career development programs and provide internships to the students, so that they students can connect with industries, and which will really help them in the future.
Yeah, funding is a big part of it, and that, of course, that's the whole point of this scholarship, is to try and help in our own small way, and to try and help people like you to be able to continue your work, because it's really, really important. And Sheila, how about you? What's the biggest challenge that you face, and how can our industry assist with that?
Yeah, I also agree, like with Laura and Rajbir. And like besides that, in my project, like I'm work with X-ray image. So the challenge part for me, it is because beside my plant pathology, the genetic studies, I had to conduct like X-ray scanning to learn like image analysis and data science. And I had to work like with computer scientists and people and scientists from the Canadian light sort. So for me, how the industry can help, they can help with their expertise and help with technology. Like if we share the technology, maybe we can bridge the gap between plant science, plant pathology, and image analysis or computer science. So I think like besides all that the Laura and Rajbir said, I face like this kind of problem, and the industry, they may have like an important role if they can share like expertise and technology.
Now, the three of you aren't going to be students forever, obviously. I'm curious to know about the future. What excites you the most about your field, about your research, and where you'd like to go in the future? I'll start with Sheila this time. Sheila, what excites you most when you think about your future career in this industry?
Yeah, like what excites me most, it is to help like improve the genetic resistance to Fusarium head blight, and also to improve like food safety. As I mentioned before, like Fusarium head blight, it is an important disease, and we have the mycotoxins, we have the problems with the damaged kernels. So what excites me most, it is the possibility to help the farmers, like to identify these kind of problems, and if we improve the phenotyping, we can also improve like the selection of resistant germplasm and consequently improve like the food safety and wheat quality.
And Rajbir, how about you? What excites you the most when you think about the future of your discipline? Maybe something you'd like to do in the future once you're finished school?
I think for everyone who is in plant science, their main focus or the main motive of their research or projects is to help farmers. Like ultimately to uh, help farmers and support them in any way possible. So I would love to contribute in future as well to the agriculture sector. I would love to develop new resilient crops which will be resistance to biotic stresses and which will have more nutritional values. So that's it.
And Laura, how about you? What excites you the most when you think about the future and what's coming after school?
Really, what excites me the most about being in plant science is seeing all this development of new technologies that we can incorporate into our field. Like since the beginning of, for example, I really want to explore more the disease management aspect of the plant science in general. And I'm really excited to know how the technologies are bringing more tools. For example, since the beginning of a disease spot, all these monitoring tools and going all the way to identification of all of these diseases. That really excites me a lot. That even when some people could consider plant science and minimize this field as just agriculture, I'm really excited to like make more out of the tools that we have available, to be more of a rounded scientist, to be able to use bioinformatics, to make use of different areas to like bring more exciting research into this field.
Now, before I let each of you go, I want to just talk about a little bit about mentorship and being masters and PhD students. I'm curious what you think makes somebody successful in plant science. So people who are just beginning their studies, just entering university, or maybe they're in high school and they're thinking of entering plant science. What's your advice for what makes a successful plant scientist? I'll start with Rajbir this time. What would be your advice to them, Rajbir?
Um, I think first of all, it's very important to connect to your roots, to your soil, and it's important to understand the challenges that the farmers and the local farmers face, and to talk with them, like what they do and what the students can do to help them. And I guess the high school students or the students who are just in their undergrads, they should, if they really want to get into plant science, they should get a good knowledge about agriculture and science and the different disciplines like agronomy, pathology, and biotechnology. This will really help them to get a better knowledge. And I also believe that they should read a lot of literature that will really help them. Also, seeking opportunities to work in the laboratories and field, will, especially volunteering, that would really help them to get better in um, pro, better graduate programs in the future.
And what about you, Laura? What would your advice be?
Mm, I would say, first of all, find a project that you feel passionate about, and also try to make as much networking as you can, because that's really important. Try to connect with people who are working in your field of study, the field that you're interested in. And down the road, when you're into plant science, you are sure gonna face some hurdles. So when they come down, just try to keep in mind that you're studying one of the most exciting fields. Plant science is a really exciting field, and don't hesitate to contact people who are experts in the field, as they're always willing to help out. At least that has been my experience, and I think that's something that students have to keep in mind as well, that you're not alone in this journey. And that's pretty much how you enjoy this road.
Sheila, I'll give you the final word before we sign off. What would your advice be to a young person just starting out, thinking about being a plant scientist, for how they can be successful doing this?
I agree with Rajbir and Laura. And I also would say, like, you need to be persistent because many challenges will arise along the way, and you need to persevere. And also, like, for me, you need to be passionate with your project, with your area of research, because everything it's easier when you like what you do. So I think that's it. Like, are my advices.
Yeah, it's important to like what you do, otherwise it's no fun to go to work every day. So that's great advice indeed. And no, thank you everybody. This has been a great conversation. And congratulations on being 2024 Canadian Plant Breeding Innovation Scholars, and thank you for joining us on Seed Speaks today.
Thank you for the opportunity, and thank you for all the sponsors for the scholarship.
Thanks so much, everyone. And I just want to take one moment again to thank our sponsors of the scholarship this year: Alberta Grains, Canadian Seed Growers Association, FP Genetics, High Tech Production, Richardson, Sasqueet, Secan, Seeds Canada, Seed World Canada, Warburtons, and the Western Grains Research Foundation. Thank you very much. And this is Mark Zinkowitz of Seed World Group signing off for the day. Thank you to our audience for spending part of your lunch hour with us. And thank you to our 3 scholarship recipients for being a part of today's episode. Again, this is Mark Zinkowitz signing off, and have a great day, everybody.